In the last video, we saw that GABA is the main inhibitory neurotransmitter of the mammalian brain. GABA acts via two distinct mechanisms. At the synapse, GABA activates GABA-A receptors, ligand-gated ion channels permeable to chloride, and here it drives a hyperpolarization. GABA also acts extrasynaptically upon GABA-B receptors. Here, the GABA-B receptor is a 7-transmembrane protein. It's metabotropic. It activates G proteins, and downstream of that, it activates a potassium conductance, also causing hyperpolarization. So there are two distinct mechanisms for GABA inhibition, and in this video, we're going to take a closer look at the GABA-A mediated inhibition. The GABA-A conductance is a chloride conductance. Chloride reverses at around minus 80 millivolts, and so typically when a presynaptic GABAergic neuron fires an action potential, it propagates down its axon, releases GABA, it acts upon GABA-A receptors, and causes a hyperpolarization in the postsynaptic neuron. That's the fast hyperpolarizing action of the GABA-A receptor. In this lesson, we're going to look in more details at the fast inhibition mediated by GABA-A conductances and see a little bit more about the specific details of how it works. The GABA-A receptor is a chloride channel, and GABA binding to the GABA receptor increases the open probability of the receptor. And so in the absence of GABA, if we are recording with a electrode looking at the single channel, then the openings and closings of this receptor, it'll be closed. If then you add GABA to the recording pipette, you'll see that the open probability increases, and we have single channel currents that come with a conductance of something like 10 to 30 picosiemens. So GABA increases the open probability of the GABA-A receptor. It's of course also interesting to know what the time course of activation of the GABA currents are. And in particular, when we're thinking about synaptic release of GABA, we're thinking about brief pulses of GABA that maybe reach 1 millimolar concentrations. And so in the same way that we thought about analyzing the AMPA receptors, we can also record the currents evoked by brief GABA applications onto patches of membrane containing the GABA receptor. And so we can have our laminar flow electrode, which has either no GABA or contains maybe 1 millimolar GABA. We mount this onto a piezo device, and we can then give brief 1 millisecond pulses of GABA to our patch of membrane containing GABA-A receptors. And we can then measure the resulting current under voltage clamp. So we keep the membrane potential clamped, and we simply measure the current flowing through the GABA receptor. And here you can see a typical recording like this. 1 millisecond pulse of GABA almost immediately activates the GABA conductance, increased open probability, chloride flux, and we get an outward current. The GABA disappears, it's only a 1 millisecond pulse, but the current remains on for some 10 to 20 milliseconds. That's the duration of the GABAergic conductance that's mediated by a single 1 millisecond pulse of GABA. So it's a fast conductance that's mediated by the GABA-A receptor, but it's interesting to note that this is still much slower than the AMPA currents that are mediated by glutamate at glutamatergic synapses. And so if you do the equivalent experiment, and last week this is what we looked at, 1 millisecond pulse of glutamate activate an AMPA conductance, and that lasts maybe only 2 milliseconds. And so the GABAergic conductance here is 5 to 10 times slower than the AMPA conductance of the glutamatergic synapses. So GABAergic inhibition is fast, but it's much slower than the AMPA-mediated excitation. Let's see how those conductances then are reflected in postsynaptic membrane potential changes. So the GABA currents are chloride currents. They therefore reverse at the chloride reversal potential, something like minus 75 millivolts. The GABA conductance is basically linear, and so we can draw a linear current voltage relationship. So this is the current evoked by a GABA pulse, and this is the membrane potential at which that current is being recorded. The equivalent ampere current would look something like this, where a pulse of glutamate will cause a current, and here it reverses close to 0 millivolts. For GABA, it reverses close to minus 75 millivolts, and the interesting range of membrane potentials physiologically is something around minus 75 to minus 40 millivolts. That's where the membrane potential of a neuron typically is. And here we have an outward current for GABA, and we have inward currents for AMPA, so inhibition and excitation. If we now think about the synaptic situation, where we might imagine having a tight, small cell that we can consider as isopotential. We have a GABAergic synapse that's made here. An action potential invades, releases GABA, and activates the GABA-A currents. And we then measure here in voltage clamp mode, we measure the current flowing through the GABA receptor. The action potential causes release of GABA, and we can then see the synaptic conductances here at different membrane potentials. At minus 100 millivolts, we have an inward current. At minus 75 millivolts, no current flow. And at positive potentials to the reversal potential, say at minus 50 millivolts, we have an outward current. And it's linear and symmetrical around the reversal potential, with the time course of around 10 milliseconds, just as we saw for the isolated receptor with the 1 millisecond pulse of GABA. So that's the synaptic GABA conductance that tries to bring the membrane potential to around minus 75 millivolts and has a time course of some 10 to 20 milliseconds. If we now think about the situation in current clamp mode, where we're now measuring the membrane potential, and we let the membrane potential swing freely depending upon the conductances that are activated, we see that there's an inhibitory postsynaptic potential that has a time course of a little bit longer than the GABA conductance, the GABA currents themselves. And that's because, of course, we have the membrane time constants that we need to discharge the capacitor. After we've charged with our currents, we need to think about there's some additional filtering because of the capacitance of real cells. Now, in this case, when we're thinking about the GABAergic conductance, I've indicated that we're adding 100 picoampere to the current of the cell that we're recording from. So at resting membrane potentials, maybe we're sitting close to the reversal potential for chloride at minus 75 millivolts. And so if we stimulate an action potential in the presynaptic cell and the postsynaptic cell is at minus 75 millivolts, we're not going to see a response. We're here at the reversal potential. There's no current flow, and so there's no change in membrane potential. We need to depolarize the cell in order to see the inhibitory postsynaptic potentials. And we can do that by injecting 100 picoamperes of current. And so here, if we inject 100 picoampere of current, we'll charge the membrane of the postsynaptic cell. Maybe we depolarize it to minus 50 millivolts. And now, when we stimulate an action potential in the presynaptic cell, we get the hyperpolarizing inhibitory postsynaptic potential that lasts some tens of milliseconds. Now, it's interesting to note that even though the membrane potential of cells is often close to the chloride reversal potential, and therefore you actually don't record an inhibitory postsynaptic potential when GABA is released, there's nonetheless an impact upon the integration of the postsynaptic cell in terms of, say, how excitatory conductances are integrated in that neuron. And that comes about because there's a change in the overall membrane resistance. And I'll illustrate that with a simple example here. We can imagine, for example, a glutamatergic excitatory conductance coming into the dendrites of a cell. That then causes current flow into the dendrite that will then go towards the soma. And so there'll be conductances from glutamate that mediate currents, charge flow via the AMPA and NMDA receptors that contribute to changing the membrane potential of the soma. At the soma, we can imagine that there are GABAergic synapses that are present here, a chloride conductance that maybe is reversing at exactly the same as the membrane potential of that cell. And nonetheless, activating this GABA conductance will change the impact of that glutamate. Why is that? Let's think about the electrical equivalent of the cell soma. So we have current coming into the soma from the dendrites. That'll contribute to charging the membrane, the capacitance of the cell, and also, of course, some of that current will flow across the leak conductances that are present in the plasma membrane. And that current, of course, in the end, goes out into the extracellular space where there's 0 millivolt potential by definition. So we have a capacitor of the soma, and we have the resistance of the soma. And what we can do with the GABAergic conductance then is to add on an additional conducting pathway here. So the GABAergic conductor here can then be added, and it changes the total resistance of the soma. And by changing the so-called input resistance of the soma, that then changes the integrative properties of how currents flowing into that soma affect membrane potential. We can think about the equations that govern the membrane potential changes here that we've already dealt with in our previous weeks of study. So the potential here across the cell is equal to the current flow times the resistance, and then we have the time course for those changes that are the charging of the capacitor here. And the membrane time constant is equal to the resistance times the capacitance. That's the exponential charging function here. And so if we have a square current pulse flowing into the cell soma, that causes a change in membrane potential, charging of the capacitor, you turn the current off, and the capacitor discharges. So this is the current, and this is the voltage. Now let's imagine that we put the GABAergic inhibition on. The change here is a decrease in the membrane resistance. We've added more conductance to the membrane of the soma, and it's reversing at the same as the resting membrane potential of the cell. So we can simply just think of it as a change in the resistance of the membrane. So we decrease the resistance of that membrane. That has two impacts if we look at this equation here. The first is that the steady state voltage is going to become less because that depends upon the input resistance of the cell. And the other impact is that the membrane time constant is going to change. It's going to decrease. So we've got a decrease in the membrane time constant. And if we now look at how the membrane potential will change, it's faster. So the overall time constant here is faster than it was, but also the overall change in membrane potential is smaller. And so by activating a GABAergic conductance, even if it's at the same membrane potential as the actual cell that we're recording from, and there's no inhibitory postsynaptic potential per se, that'll still cause an inhibition of excitatory glutamatergic inputs coming in. And so here we thought about steady state injections of currents, but we'll have the same situation if we think about glutamatergic inputs coming in, brief currents, where if we have at resting conditions an EPSP that looks like this, then we turn on the GABAergic conductance and the EPSP gets smaller and also a bit faster because we've changed the membrane time constant and the input resistance of the cell soma. So shunting inhibition is another important form of inhibition in addition to the hyperpolarizing inhibition mediated by charge flowing through the GABA receptor. Now, GABAergic synapses are made on a large variety of subcellular compartments. GABAergic inhibition can occur here on the distal dendrites, so we have GABAergic input right here in the distal dendritic arborizations, and presumably a GABAergic input here will have a large effect upon the nearby excitatory synapses coming in here, but might not affect GABAergic synapses occurring on other dendritic branches. There might be specific inhibition that's localized to individual dendritic arborizations. So there could be high degree of spatial specificity as to where the GABAergic inhibition might function. There are also GABAergic inhibitory synapses all over the basal and more proximal dendrites, and interestingly, there are also GABAergic synapses that are directly placed on the soma. And that's something that you won't find usually for excitatory glutamatergic synapses. They're sitting on the dendrites, but not on the soma. So GABAergic inhibition is different and prominent on the cell bodies, where it could affect the input resistance and the integration of the synaptic currents flowing in from the dendrites in this divisive way by changing the input resistance of the cell soma. Interestingly, there are also GABAergic synapses directly on the axon initial segment. Again, there are no excitatory synapses in this area, but inhibitory synapses are prominent here at this site here where action potentials are initiated in most cells. And GABAergic inhibition here then might not affect dendritic integration at all, but might simply form an editing function where it decides whether a cell can output an action potential or not, independent of what synaptic computations are occurring in the rest of the cell. And finally, in the spinal cord, but not anywhere else in the mammalian brain, there are also GABA-A synapses directly here on the presynaptic specializations, and that only occurs in the spinal cord. But these synapses at other locations occur throughout the mammalian brain, and each one probably performs a slightly different function. So in this video, we've seen that the GABA-A conductance lasts some 10 to 20 milliseconds and has a reversal potential of somewhere around minus 75 millivolts, depending upon the local chloride concentrations. The GABA-A conductance can mediate inhibition in two slightly different ways, both of which, of course, depend upon the chloride conductance mediated by the ligand-gated GABA-A receptor. The first one is hyperpolarizing inhibition, and this occurs when the membrane potential is depolarized relative to the chloride reversal potential. And that's simply a movement then of chloride ions in, negative charge inside the cell, causing a change in membrane potential. The other sort of inhibition relies upon a conductance and a change in the input resistance of a cell. And so even if the membrane potential is actually sitting at the same equilibrium potential as for chloride, there can still be a significant impact upon the integration of excitatory inputs. That ligand-gated A chloride conductance will decrease the input resistance of the soma, and the excitatory synaptic input that arrives at the soma then causes a smaller change in membrane potential if the GABA-A conductance is open. So there are multiple different ways in which GABA can cause inhibition, and furthermore, it's complicated by the spatial distribution of the GABA-A input that'll presumably have many different effects depending upon where exactly the GABAergic synapses are activated. And we'll learn more about the subcellular targeting of GABA synapses in subsequent videos.